So this is meant to be more of a kind of discussion anyways. There is more material than I could, kind of put into this little summary. So I'm actually focusing on just a couple of the aspects that they talked about today. So there were all kinds of, single cell technologies, but I wanted to focus on kind of these micro-physiological systems, which come in a couple flavors. So I'm just gonna switch over, but this is a small group, so let's kind of keep it colloquial. Let me share my screen. So, all right. Hello, Rajev. So can you guys all see my screen?
Yeah.
Okay, great. Let me actually turn on the camera here. Okay. So, yeah, just following up on these conferences that took place this week. So first there was a microfluidics conference, and then this organoids conference, which is called 2D to 3D/organoid. So there is a lot of biology in all those, actually more than technology, even at the microfluidics conference. I think that's a sign of maturity of the field. So there's a few maybe areas that one could split it into. So single cells and droplets can be, related, but they have somewhat different, overlap. I have great interest in functional assays, and I could mention that, but I thought I would keep it all off for now. And then even some more tools that are kind of a little bit more specific to disease rather than this issue of biomimicry and vascularization. So vascularization comes essentially in two ways in these microphysiological systems. There is sort of this top-down approach with 3D bioprinting. And I have started to put the names down, but I haven't finished it. So this is just a sampling. Then there are the bottom-up approaches where you start with induced pluripotent stem cells, typically let them differentiate and self-assemble. So there's a number of people working on that. And then there's a few hybrid approaches. Yeah, if you can interrupt anytime on this one. So just to kind of show you some examples of what people are up to. So the slides will kind of have this classification of the approach and something about the person and their affiliation. So Roger Kam has been a luminary in this field for a long time. He is interested in organoids for cancer and brain and microphysiological systems. He's a co-founder of this company, AIM Biotech. It's in Singapore, where they had a lab. So, okay, we got a couple more people. So this device is not only used for neural growth, but also for vascularization. So the design of it.
Probably not, but it could need to charge us a little bit.
Hello?
Yeah.
Okay. So here's the schematic of the device. There is a central gel channel that's separated with these posts, and they actually have triangular posts, which helps with proper pinning of the meniscus. And so here's a use, and you can see, I believe these are also early stages of neural growth. But that's what they use a lot in these systems, but also you can impose vasculature in this middle gel if you have the right type of cells. And they've studied many different types of cells, specifically, for example, for the blood-brain barrier model. They're using 3 cell types: endothelial, astrocytes, and pericytes. And they create this barrier, and they measure the so-called tear function. So that's a sign of permeability, and you can measure it. Traditionally, it's measured with electrophysiology, but you can also do that as a proxy with dextran with fluorophores. So some of the observations are that this 3D constructs, they improve the strength of the barrier, which is very important for being able to study disruption of it with drugs. And some of the questions they're asking is, why are some of the cells able to metastasize across this very tight interface with tight junctions? And that process is called extravasation, because essentially they are crossing this barrier from a vein and into the tissue, which is brain tissue there. So, for example, they found that some of the cytokines, signaling cytokines, this protein CCL2 and interleukin 8, are increased in one particular cell type, the astrocytes versus the endothelial cells. Pericytes are kind of on the outside supporting it all. And here are a few examples of some collaborations and people who are using it. So they're working with Amgen to study this vascular permeability, and with Biogen they're working on a couple of neurodegenerative models. In the Lou Gehrig's disease, this ALS model, they're looking also at some functional assays, which is kind of a specialty of this company called Hesperus out of South Carolina, with a presentation that was given by Jay Hickman, but I'm not going to go over that. It's just this one connection to the functional assay here. Yes, so Jay was actually also looking at neurodegenerative models, found some very interesting things, but he is finding impact on the muscle cells, but specifically his main hypothesis is the neuromuscular junction that's driving this disease. And it's been, probably they call it the area with the most publications with no Nobel Prize, and also probably the one with the most failed drugs. So they're also working on Alzheimer's with MGH and its interaction with vasculature. So there are impacts on other cell types, such as this protein downregulated in endothelial cells. So one of the questions I was interested in was the impact of other cells which are not present in this list, of the smooth muscle cells. So Roger's answer to that was that they're important for larger constructs, but not for this microvasculature. So if you guys are okay with this, I'll kind of keep going, but please interrupt me. Or I'll try to keep an eye on the texts as well. Okay, so hopefully I won't miss anything. So some more examples from bottom up. So for example, from University of Delaware, John Slater was reporting on encapsulation of hydrogels in synthetic gels with different stiffness. So this is another kind of feature here, the impact of the matrix, and including its physical properties as well as chemical properties. So cells in the matrix, which is smaller than cell size, can't easily get through, so they begin to degrade it. And this is an important process in metastasis, obviously. So they use, for example, some highly invasive triple-negative breast cancer cell lines to do these studies. So they synchronize them in G0 phase, they encapsulate these cells to form spheroids or organoids, and they do that in these combination matrices with 16, typically by 4x4 effects, to study stiffness and also adhesion points. So these are the two parameters with 4 different gradations, 4 of stiffness and 4 of adhesion points. So here's a couple of observations. A lack of adhesion points leads to cell death. So this is the mechanism by which detached circulating tumor cells are supposed to undergo apoptosis. So that's the assumption here, but they certainly see that majority of them are undergoing apoptosis by presence of caspases. The maximum survival is seen in matrices with this combination of features. Not excessively high adhesion, not too low, but degradability is high. So apparently they may prefer softer structures, which seems to be kind of general case, although it varies obviously by organ type. They've also studied single cells and clusters. And you can regulate the cell cancerogenic nature by hydrogel properties. For example, one of the things in these cells that do not apoptose but go into a dormant state is their ability to be reactivated when they're given a better matrix. And this could be achieved by a few mechanisms, but it's physically provided. So these cells actually become reactivated and begin replicating. So this is, a mechanism for greater metastasis. And also, they've done a lot of modeling, so they kind of have some rules for these matrices, but also they're working on perfusion of these structures because it is limiting oftentimes. Another example from Peter Ertl in University of Medicine in Vienna was for neurodegenerative Parkinson on a chip. So they're also going for increased relevance of these in vitro models. And specifically, they're combining induced pluripotent stem cells with organ on the chip. And they're testing two kind of primary preclinical endpoints, toxicity and efficacy. So Parkinson's is characterized by a few features, notably loss of dopamine receptors and accumulation of a protein that forms aggregates, like is often the case in neurodegenerative diseases. So the model is organoid midbrain. It requires a lot of oxygen, so they integrated optical sensors. The flow rate is important, but it's actually quite low, so this is a linear flow rate. Interestingly enough, neurons align with flow, and the flow has been shown to significantly reduce necrotic core on day 35, which is actually very favorable in a lot of ways because it's difficult to achieve this anyway. So it's actually rather remarkable that these dense organoids have so little core. And the endpoints were measurement of dopamine in situ and actual electrochemistry of secretion or the electrochemical, the electrophysiological signal. Electrochemistry specifically to detect dopamine had to be made selective against ascorbic acid, and they used self-assembled monolayers on gold to achieve that selectivity, which was actually quite good. It was missing some of the other metabolites, but as they found, they were not present in these models. So in this model, their relatively simple electrochemistry setup was selective for dopamine. So it's kind of an example of showing it, but it's interesting that there are more and more integrations of sensors and kind of functional assessment. So this is one of the more top-down kind of approaches where the physiology is put together in a macro scale. And so they were studying here locally at BU issue of a matrix and also vascularization for tissue engineering. And one of the applications is this heart patch, which is a relatively common congenital disease in infants. Unfortunately, the homografts, from the individual even have they have no immune rejection, but they have other problems, aneurysms, problems with contraction, and also excessive calcification. There are several tissue layers from the inner ones to the medium to the fur ones, the intima media and adventitia. And they can be printed in various gels and then layered. So one of the techniques is to use alginate and cross-link it with harsh reddish peroxidase in the presence of this metabolite, which is a little bit dopamine-like and is able to polymerize apparently with some radical chemistry. So it's important to study physical properties of these tissue sheets, but they're extremely fragile. So they've used a very interesting method. They've embedded magnetic spheres into these sheets and used magnetic Hall effect to measure the stretching, because otherwise it has, the Young's modulus of toilet paper. So it's virtually impossible to stretch in any traditional instrument. And here are some specifics of what they were doing. They're focusing on collagen type 1, which is the most common one, but there are 17 types in the body, which kind of shows the prevalence of different matrix types and importance. There is also importance of alignment, which can be studied by X-rays, and there's some other studies that talk about this. Importantly, there are techniques using microfluidics, such as shear flow, elongation, and kind of a flow focusing. So they used these techniques to make matrices using a variety of proteins, including silk proteins, which have a prevalence for that type of aggregation, and to study the impact of mechanical properties on the cells and degradation of that network. So let me mention a few more. So this is more of an application, a kind of example. This a company is printing induced pluripotent cells using a variety of gels with this 3D cell printer. This company was a sponsor, and they're focusing on a couple applications, including using cells for diagnostics, which is very interesting. So how to control that? Well, you print them in gels, and as I mentioned, some of them could be used for immuno-oncology diagnostic applications, predicting patient response. And what they've shown is that these 3D organoids are better at inducing T-cell differentiation. So this is being applied by them for their own drug development. This researcher from the University of Washington was also applying a 3D printed approach using, in this case, not collagen, but more controlled chemistry. So this is polyethylene glycol, a spacer of various molecular weight, with diacrylate moieties on both ends. And it's an alternative for microfluidics to PDMS. And one can print organ-on-a-chip biology inside these devices. And certainly the best applications for these printed technologies are where the devices would be hard to print. Sometimes they use photopolymerization to build them up layer by layer. There are other mechanisms, but this is one they often use. And molecular weight of the spacer controls porosity and stiffness. So they're kind of using this digital manufacturing using digital light projection from Texas Instruments. Unfortunately, it's not the highest priority for this company, so they're not very responsive for adjusting their technology to these needs. So some of their instruments still have rather small resolution. Alternative technologies, such as from Boston Microfluidics, are able to achieve higher resolution, although they're a little bit more cumbersome. In comparison, PDMS with photolithography goes down to a micron resolution, but suffers from problems of process of production. It's very manual, as well as materials. It's rather sticky for proteins, not like cyclic olefin copolymer, which is low absorption. And also, it's very porous absorbing drugs. So this is terrible for low concentration drugs. So the 3D printing can be cheaper and make complex devices. So in addition, there are labs that do this as a service. But while the transparency is good, their biocompatibility is poor. And some of these, particularly the smaller molecular weight cross-linkers, are actually shown to be non-cytotoxic, if you properly treat the construct afterwards. So it's another illustration of the top-down technology with more kind of chemistry control. So this is from CellLink themselves, a gentleman named Patrick Thayer, who was talking about a few applications. Specifically, drug development obviously has a very problematic process and very long development cycle, but also organ replacement. And these are all very expensive processes. but it's not the only ones right now that's driving it. Performance matters as much as costs. So what selling provides is a variety of materials, like PLA, PCL, some biodegradable materials. They also have a variety of syringes or nozzles to dispense it. And now they've started to produce a variety of formulations or inks for printing different cell types. So cells from primary cultures or from organoids could be dispensed and printed in this fashion, and it is quite sensitive, obviously. So they have now quite a variety of instruments. One of them with higher resolution is using photo cross-linking, and it's often used to print organ on a chip. They also have tools for droplet on demand and single cell dispensing and select cell imaging. So this is kind of a corporate approach, I guess. But they're also obviously concerned with the biology of these interactions of the cells and materials, because that's their core technology. Some of the applications of these include making spheroids such as for liver, which is a very important organ for toxicology. And they often use relatively basic assays for that because they're more a technology company, such as LiveDat or accumulation of lipids. In CAR T technology, in addition to DEF, they were also kind of observing the T cells visually, but it is still a relatively late endpoint. And obviously, one of the implications of all of this is that they've also been seeing evidence that two-dimensional cultures do not recapitulate 2D. And Patrick provided examples both ways, actually, where 2D provides false positives and false negatives in terms of these effects. So let me see if, okay, it looks like I can keep going for a moment. So let me tell you about another a bit of work from Northeastern by Carol Livermore, who is doing a very creative approach of taking these structures and not just layering them, but doing it in a very creative way to create liver on a chip. So this is supposed to say liver. So liver has several substructures. There are lobules and sinusoids. And sinusoids are characterized by essentially a channel that's lined with endothelial cells on the inside and hepatocytes on the outside. And origami is able to produce that type of transition from 2D to 3D. And they made repeated structures using initially fan type of assembly, but then they made more complicated multi-layered structures. The impressive thing is that these very thin polycarbonate membranes are quite robust and surprisingly easy to handle. I actually inquired about the facility of this, and so it's apparently not a problem. So they started with kind of a simple fan design, which they cross-linked. Now they have more of an external packaging, which keeps things intact. And their applications are drug development or personalized medicine. So this is a kind of ex vivo application. And they have also demonstrated exciting functional outputs, which I think is an excellent sign of maturity, again, of the field, looking at output, of the whole structure, which is producing albumin and urea, just like in vivo. So let me give you a few more examples, and then I would like to stop and just discuss questions with you guys. So Mario was reporting on organ on the chip tools for orthopedic research. So they're making a number of joints, looking a lot at cartilage type of materials. So the challenge, as he was also pointing out, is to have a proper balance of features that are tractable and relevant. And also on kind of the biology side, this is technical. The biology side, it's more of a organ on a chip versus perhaps lab on a chip, where organ on a chip is all about growth, whereas lab on a chip is more about sensors. But in fact, one could combine and balance the two. So here's a few examples of what they were studying with some interesting observations. So for example, they made a lab on a chip from the synovial joint. They stimulated it with TNF, not THF, TNF alpha, tumor necrosis factor. And also they were observing that organoids were in fact much more homogeneous when grown in this fashion. And so they were able to, certainly better explain the effects of these stimulants. They also have exciting multi kind of level cartilage chips, and they're using a large animal. So this is a horse model, but it mimics, human physiology pretty well. Lots of, need to carry weight. One of the things that they were noting is that there are gradients that naturally are created within these systems. So they were trying to replicate them by putting few of these cultures on the chip. So in particular, they grew chondrocytes on the chip and studied their inflammation, but then they combined them with cartilage, this synovial tissue on opposite sides of the channel, and they were studying inflammation. And they saw that there is asymmetry. So inflammation triggered on the synovial side would propagate to the other side, but not vice versa. So there is a lot of nuance that's being shown in this combination, but it's probably requiring these large assemblies. It couldn't be done with simpler two-dimensional cultures, perhaps. So as I was mentioning, these are truly three-dimensional cultures, and the height of the channel for that reason is selected to be quite tall. So this is kind of mesofluidic cultures almost, the large organoids. Some more observations. Passage numbers matters. Typically, it's best to use primary cells. Of course, it's very limiting. Specifically, the problem with a lot of current media, which many people are aware of, and for example, James Hickman, that I mentioned, professor from UCF, has for 25 years banned use of fetal bovine serum in his media. In fact, they use only synthetic media because they are so concerned about signaling molecules in this uncontrolled media. Specifically, FBS is known to cause dedifferentiation, which is very important when you're working with organ on the chip, kind of induced pluripotent stem cells. So matrigel, which shrinks just like fibrin on some of these co-cultures, which is natural, has some of the same kind of effects that FBS is prone to. It's made from a very kind of a crazy mouse sarcoma line. So it's a very interesting and troublesome material, but it is kind of the gold standard extracellular matrix at the moment, despite the lack of reproducibility and storage problems. And Mario was even pointing out that not only is it bad lot to lot, but even within lot. So it kind of, pays to work with what you've got and go slow. So let me show a couple more examples. So this is a company called Fluid Form, and Andrew Lee was reporting on their technology of printing inside a gel. So it's formation inside fluids, and basically what they're trying to do is to help to close this gap between structure and function. As I was saying, they're kind of different angles people are coming at it, and also assaying more and more at the functional level. And it requires softer materials and control of geometries, and they've seen impact of geometries on vascularization, differentiation, everything. So what they do is have a sacrificial gel, and actually the needle penetrates inside this gel and deposits some material. Material could be one of these 4 types they currently do, which can be cross-linked in different ways. And they've demonstrated at least one example with their logo of all 4 types being used. So there is some ability to combine them. What they do a lot of the times is print from collagen 1 rather large structures. So for example, they would print things like tripartite heart valve that opens kind of unidirectionally. But it's a very important structure for understanding congenital heart disease. Um, um, and in addition to kind of mechanistic studies, they also were able to monitor, oops, this should say waves, calcium waves in cells. Now, these cells are plated in pretty high density, but despite calcium waves, they're not contracting very much. So my assessment at the moment is these are better models at the functional level of the mechanistic structure than the physiological contraction. And they were also studying these cells using metabolism as well as observation of morphology. So I think I'm getting to the hybrid methods pretty soon, but maybe just one more from top down. So this was a laser-assisted bioprinting from a company out of France. They print a large-scale tissues, and they're focusing on being able to do this reproducibly with standardization and large-scale bioprinting. So it's also trying to close some of the same gap that I've mentioned before between structure and function for organ regeneration among other applications. So some of the things that they're concerned about is viability based on the shear in the nozzles, but they are kind of also revolutionizing next generation bioprinting because this laser-assisted method can have a high resolution, and they're actually able to achieve a very high viability. So I guess one more, I spoke too soon, about the top-down. So this is an example of a bone model. So they're trying to make bone structure and then vascularize it. So there is a structure that looks like a wrap with a blood vessel in the middle, and kind of layers of bone woven around it. So that's one of the unit that's structured. And it takes weeks to lay down this matrix layer by layer, and then they wrap it. And the layers are pretty thin. After growing them for about a month, they do studies such as scratch and monitor. And this is a very common assay. There were others reporting on improvements of the wound healing assay with microfluidics. So this is bone healing. On the material side, the addition of polyvinyl alcohol can stiffen. Oh, sorry, that's a typo. Can stiffen and laminate this piece of gel, and PVA can be washed out. But this is obviously something that requires a very careful control, because it can be I mean, the high molecular weight is probably not so cytotoxic, but it's more difficult to wash out. So there can be residual effects of that additive, and also, of course, the stiffness, as we were saying, matters. And also, they noted that for bone growth, the role of hypoxia in these larger structures is important. So a few examples of hybrid methods. So from Northeastern, Professor Dye was talking about a brain model glioblastoma. These are extremely invasive but orphan diseases. Now, these glioblastoma cells can be injected, xenografted into the mouse brain, either as single cells or as spheroids or organoids. Spheroids are smaller than organoids, and they can also be studied on chip. There are a couple of endpoints that are possible. One could do a single cell analysis or kind of aggregate endpoints. They, in the chip examples, they kind of bioprint some channels to help with this production of channels. An interesting observation that they've observed, and this is one of the key kind of points about vascularization of interest to me personally, is how to produce connections of artificial microfluidic channels with vasculature. And they, as some others, have noted that in the early stages of vascularization, flow actually suppresses sprouting of endothelial cells that starts angiogenesis. So they can be seen by staining this Z01 protein, and it's done in 3D using printed capillaries. And the assembly can also be done in sheets, as well as 3D printing. But the ability to control these layers also allows customization of microenvironment with additives in terms of stiffness. And one of their future goals is to study extracellular matrix. I asked about their latest device, Model B. How long does it take them to create their devices? And they admit that it's rather long and technically challenging. So this is kind of characteristic of the field. It's still rather an art. They're able to achieve a lot of technical solutions, but kind of in-house. It's not easily kitted. And some of the other presenters also made that same point, that it is the need to communicate requirements to manufacturers and produce designs fit for purpose that is a challenge. And so this is kind of an example of the need for these researchers to do both. So hopefully we'll be able to build some better tools for them. An interesting hybrid approach is with kind of needling these organoids by Dr. Hibino. So they're using this to generate heart tissue and study heart failure. So they generate spheroids, and then they physically put them on this needle array. So there is no chemicals or cross-linking, there is no biomaterial. So this is very kind of pure in terms of the bottom up, but top down, they're able to put pretty large volumes together in this fashion using a combination of cell types. And this is a good chunk of tissue that is suitable for implantation. But there are some oxygenation problems and necrosis, not as grave as one might expect, because as you can see, there are some interstitial spaces. So there is a little bit of perfusion that's perhaps possible. And challenges are kind of exactly what you might expect. They are looking to increase to extend maturation of these tissues. One of the things that has been observed with single cell technologies is that people generally get early stage differentiated cells. It's hard to get late stage. Also, that late stage and mass of biomaterial requires vascularization to avoid necrosis. And finally, specific to their biomodel, the vascularization needs to be controlled because they wish to study contractile function, which could be adversely impacted by too much vasculature. So another hybrid example for disease modeling in chronic liver disease, very interesting, again, kind of bio-inspired. They simply decellularize a liver from a rat or a mouse. So this keeps all the vasculature and the extracellular matrix intact. And when they perfuse the liver with cells, they self-organize on this matrix and vasculature into hepatic and biliary ducts. And this takes a while. Now, some of the things they're studying are Alagille syndrome, which has mutation in a particular gene, and that does not activate Notch upon translocation to the nucleus. The point of this is that very specific mutations can be traced to lack of bile duct activation, and they were trying to rescue this with some mechanisms or induce it. Another thing of interest is liver fibrosis, which occurs by a few mechanisms, one of which is increasing tissue stiffness by secreted collagen by hepatic cells. So it's possible to observe this alignment not only under X-ray, but apparently there's differential staining with certain dyes. In the mesenchyme region, this dye, picrocereus red, can stain them with a somewhat different polarization. What they've observed, or rather different color, I should say, more green to red shift. So disorganized fibers are redder and are laid down by cancer cells. So this leads to apparently a more anisotropic stiffness, whereas with aligned fibers, the stiffness is isotropic. I mean, or rather non-isotropic. It's only in a particular direction, right? Whereas this disorganized fiber is stiff in any direction. Encapsulation of these organoids is also done, and they use a number of materials such as hyaluronic acid, gelatin, or PEG diacrylate. So this is with light-assisted polymerization. And their applications include ex vivo personalized medicine. It takes quite a while to screen these tumoroids. This was one of the questions from the audience. And their approach is, for a few cancers, is to screen drugs directly. It's not the exclusive method. So in my previous position in the company that was doing companion diagnostics for blood cancers, we were looking at a functional but indirect assay of mitochondrial function, which indicates propensity of cancer cells to undergo apoptosis. So it's an indirect effect, and there's still kind of a difference of opinions out there, which approach is more suitable. But this is a direct approach, and they are trying to screen primary cells, so with no passaging, to give themselves more predictive power. And their observations from 3D world are not surprising. They observe increased resistance of carcinoma cells, for example. So this is an example of how two-dimensional assays do not have translational value. Drugs that are seen to be within certain therapeutic window of efficacy may find that window shifted to higher concentrations, or kind of gone entirely due to various resistance from growth factors and other contact cell-to-cell signals within these three-dimensional organoids. Also, they've observed that stiffness of these gels and alignment of fibers alters WNT signaling and activation. So this is kind of a molecular signature. And finally, they xenographed certain organoids into rats, and study their susceptibility and screen for drugs there. Finally, they've observed both in vitro and in vivo in microfluidic channels that progression of colorectal cancer is associated with changes of extracellular matrix as cancer cells rearrange it. So it's a bit of a dichotomy, which of these comes first, but the detection of it in vitro suggests that there is some mechanism where cancer cells rearrange the matrix. But clearly there is, as we've already heard in one of the previous mentions, dormant cancer cells can be reactivated by extracellular matrix signals. So it's a two-way street. I think I'm almost done. This is actually it. So, so I would like to kind of pause here and maybe discuss some things with you guys, answer your questions.
Hey, Max.
Uh-huh.
This is Nikolai.
Sorry.
So I actually have a quick question. So all of this is good, but in my my problem with a lot of this is how to scale, right?
Yes.
This is, I mean, our topic today is personalized medicine, right? All of this 3D printing solutions are great, but you can't, you may be able to deploy all of these solutions to help, I don't know, Bill Gates to replace his liver, right? So that he could drop $50 million to print one liver. But all of them are very expensive, very slow. How do you see a solution to, take all of this information and all of this knowledge? Like, what's in your opinion will be the technology that will win in terms of cost and scalability?
Right. Challenging question to try to predict, but I'll try to take a stab at it. So I think the speed is an intrinsic problem. It takes a while to grow biomass, and some of it has to be self-organized. It can't all be just 3D printed, as in the movies. So that or self-organization takes time. It may take, even with the ability to split it into pieces. For example, the organoids that are needled. Grown in parallel. There's still going to be, some time, but I think that's acceptable. The price will come down with improved technologies and scale up of this. So I think one of the largest technical challenges is ability to control some of the differentiation and make it reproducible, but also growth of sufficiently large tissues. So these, for example, your question about an organ, like a liver transplant. So for regenerative medicine, a transplant, typically people say 10 to 30% of the biomass of a full organ is required to transplant it into a person and to have it grow in situ. So that type of biomass from liver, would still be a few cubic centimeters and would be subject to necrosis. So vascularization is kind of the biggest challenge. That's why I was kind of trying to focus on various ways of introducing it top down and bottom up. I haven't mentioned all of the talks. It's just too much, way too much, even in one summary. Very short summaries, but people print microvasculature using gels or laser ablation. So there are a number of techniques that they're trying to do. How the gel and the bottom-up versus top-down combination is going to work out, that I would hesitate to judge. But I would say that there is going to be definitely some combination of both, but certainly bottom up is inevitable, although top down accelerates assembly.
Yeah, I know these companies Cell Inc. I mean, I met them at stem cell meeting many years ago when they were just like, they didn't even have money to have a booth at the meeting. And their printer is fairly cheap. It's like $10,000 or something.
There's a little bit more expensive. They have a few versions. So the one that was demoed there with 3 nozzles was, I believe, 30 or 40,000, and they have a variety of nozzles with droplet generation, UV cross-linking, or a kind of syringe peristaltic pressure or syringe based. So they have a few options. They've actually recently apparently put a few other products on the market outside of 3D printing, such as with imaging, single spell dispensing, but that's just FYI.
Yeah. Yeah. Okay. Then I'm just trying to understand a little bit better where the whole technology is leading. It's not quite prime time to print organs and transplant it into humans. It's quite low throughput to use it in like toxicity screening and drug discovery. selling, I think it was just a cool toy so that every lab could buy one, and then they could just print, but and do things like a one-off experiments. But what's
I think
where it's going?
I think there is more acceptance now than perhaps when you last interacted with them. Um, I think, for example, for toxicity studies, there's a number of companies that are moving in that direction because it's much cheaper. And in fact, by 2030, the FDA expects to do away with animal testing at all. So there's a deadline looming, which has already been done for cosmetics. So in 2012, they forbade all animal testing of cosmetics. So now there is a lot of, in fact, two of the presenters, include among other things, they were talking about skin on a chip being used for L'Oreal. So I think for toxicity, these systems are sufficiently high throughput. And also when you begin to combine the multiple organs on the chip, you can see, for example, central nervous system toxicity, not just liver. A lot of failures are kind of baked in the cake at the preclinical level, with, for example, a CNS toxicity. So I think out of necessity, people are starting to use it more and more because it improves, it's reducing risks of the process. Certainly, the throughput is a huge problem. For toxicity, it's a little higher. For efficacy, it's downright low. Modest or moderate, to put it generously. But people acknowledge it, so there is kind of what I've heard from some folks outside of this conference, as a workflow, they have organoids, for example, for development of biomarkers or for some kind of intricate work at low throughput, and then they will take that information forward in a high throughput system.
Well, I wouldn't say, for example, for liver, generating organoids in a high throughput way is a fairly straightforward way. With all of these technologies like a hanging drop and spinner flasks, I think you can just very cheaply make a lot of mini livers.
To an extent, absolutely. But one of the places for microfluidics, to compare it to spinner flasks, hanging drops are better, but spinner flasks give pretty heterogeneous material.
Yeah.
And
well, spinner flasks I know used a lot to do mini pancreas. So for mini pancreas, I think spinner flasks are kind of standard, and yeah, hanging drops are very good for livers.
Yeah, but I think you are making kind of that point as well, that people are starting to use it for different organs. It's a little bit difficult to predict what's really going to, break the dam. I would like to think that some of the tools which we might be building will basically provide products. So right now it's an art, everybody's still kind of doing it. It's pre-industrial revolution kind of technology,
Yeah, I agree with you, because for me, I just like, for many of the biotech and pharma companies, they are moving to cut the costs, right?
Mhm.
First of all, they are hiring PhD level scientists, mostly for like, group leader roles and up. And most of the bench work is done by research assistant, and they're usually like bachelor's and master's level, right? And it's like you just tell them, okay, go do like 10 westerns, or go do this culture. And I'm just trying to what my point here is, the technology itself, it has to be very, very bulletproof, so that I know somebody with a college level education will be able to do it every day for very, very reproducibly, because
Absolutely.
they're not particularly shipping ready. I know companies who make organoids, they make this like soccer balls with like gyroscopes to ship them because they're in hanging drops, and just to ship it, it's like $5,000 just to ship, you know, one like a 96 well plate with the spheroids. So people looking for solutions how to either outsource it completely or do it in house, but very robustly, like, you know, a black box with the one button, you push it and it just spits you out 5 ml of spheroids.
Yep, absolutely. So on outsourcing, just briefly, so the Hesperus company with a human on a chip, that's kind of their entire model. They do all kinds of functional studies, and they have several organs on the chip, so that's kind of their claim to fame. And apparently, Javelin Bio, which is local here in Boston, is moving in that direction. So certainly, outsourcing is favored in a lot of ways, and there are companies out of California and, you know, elsewhere that do this. I think it is possible to make some turnkey solutions, but certainly shipping these organoids is a huge impediment for some areas like IP. You know, for example, if we were to partner with Hubrecht organoid technology, which, you know, a number of researchers are already working with because they have a lot of control of the IP. It would require shipping pre-grown organoids, which would be desirable for researchers, but very hazardous to receive. And you know, some of the professors there were sharing stories of you know, precious cargo stuck in customs for 8 days, and dry ice is gone, and of course all the cells are gone. So it's a huge problem. In part, it may have to be solved by giving people the keys to grow their own systems in-house. But yeah, I'm actually working with some folks on some shipping containers, for example, for being able to ship blood for companion diagnostics. But it's different from organoids. But it could be repurposed. You know, there would be thermostating, there would be circulation of you know, artificial blood, essentially. Did I answer your question?
Yeah, thanks.
Thank you. Well, I would love to continue this, folks, including now, but in the interest of time, I think I will kind of adjourn for now. If you guys are available any of the following weeks, I would appreciate it. So thank you for coming and listening.
Thank you.
Thanks. Bye.